I'm going to share on the topic rhizobium symbiosis. So moving to the introduction, it is a soil bacteria, and this rhizobium basically it is found in soils and are about 1 micron in length or diameter, and there are 1000 microns in the millimeter. And rhizobium has the ability to fix atmospheric nitrogen in symbiotic association with host legumes. And bacteria in environments that have high levels of nutrients may be larger than those in nutrient poor conditions, and it converts atmospheric nitrogen into ammonium, and it is recommended for chickpea, lentil, black gram, and oil seeds like soya bean, groundnut, etc. It belongs to Rhizobiaceae family and are symbiotic diazotropes. It forms a endosymbiotic association with legumes. Rhizobium is a gram-negative soil bacteria. They are non-sporing bacteria and their rod-shaped structure. Nodal formation. Steps involved in rhizobium modulation process. Attraction and multiplication of rhizobium. Attachment to the root surface, root hair curling, formation of infection thread, and nodal formation. So attachment to the root surface, root hair curling, formation of infection thread, nodal formation. Attraction and multiplications of rhizobium. So this is the recognition and attachment. Root hairs of leguminous plants release flavonoids which attract rhizobium bacteria present in the soil. In response, rhizobium produces nod factors, that are signaling molecules that initiate symbiotic communications between plants and animals. Attachment to the root surface. Exopolysaccharide produced by the rhizobium interact with the lectin produced from the plants. Exopolysaccharide helps to bind rhizobium cells to the root hair. And next one is root hair curling. It produced endolastic acid. The curling of the root hairs is attributed to endolastic acid. Therefore, rhizobia become enclosed by the root hair walls. The nod factors induce curling of the root hair. This curling trap rhizobium cells inside the root hair pocket. And the root hair curling forms a structure called shepherd crook. Formation of infection thread. A tubular structure called the infection thread forms from the root hair cell wall. The nucleus of the root hair cell guides the pathway of infection thread into the root hair. And these rhizobium cells travel through this infection thread, moving deeper into the root tissue. Then nodal formation. Infection thread continues to grow beyond the root hair cell and penetrate cortex cell. On entry into the cortical cells, infection thread branches, rhizobia are released shortly into the cortical cells. And the infection thread reaches the root cortex. The plant cells in this region undergo rapid cell division, leading to the formation of a root nodule which houses the bacteria. And the cell division of the host cells and rhizobia changes their shape to form cells called bacteroids. And bacteroid formations. Inside the nodules, rhizobium differentiate into a specialized form known as bacteroid. These bacteroid are the active nitrogen fixing forms of the bacteria. So first one is the rhizobia attached to the root hair, and second step will be an infection thread is formed through which bacteria enter root cells. And third step, bacteria change into bacteroids. Packed root cell enlarges at that time, and this bacteria enlarges will be named as bacteroids, and this enlarge the root cells, forming a nodule in the roots. So this is the example of a pea plant showing the root and the root nodules. The mechanism of nitrogen fixation. The conversion of free nitrogen into nitrogenous salt to make it available for absorption of plants. And the basic requirements for nitrogen fixation is nitrogenase enzymes and hydrogenase enzyme, protective mechanism against oxygen, ferredoxin, hydrogen releasing system or electron donor, and constant supply of energy, that is ATP, and coenzymes and cofactors, that is CoA, inorganic phosphate and magnesium Mg2+, and cobalt and molybdenum also present. Nitrogenase enzyme, it is an anaerobic conditions made up of two protein subunits. One will be iron protein or dinitrogen reductase, and another one will be molybdenum iron protein or dinitrogenase. So Fe protein reacts with ATP and reduces second subunit, which ultimately reduces nitrogen into ammonia. The reduction of nitrogen into ammonia requires 6 protons and 6 electrons. 12 moles of ATPs are required for this, and 1 pair of electron requires 4 ATP. The modified equation is nitrogen in the presence of 8 hydrogen and 8 electrons gives ammonia plus hydrogen. The hydrogen produced is catalyzed into protons and electrons by hydrogenase enzyme. Pathway of nitrogen fixation in root nodules. Glucose 6-phosphate acts as an electron donor and it is converted to phosphogluconic acid with the help of NADP and water, thus obtaining 6-phosphogluconic acid and NADPH and H plus ions. Here NADPH denotes electrons to ferredoxin and protons released ferredoxin is reduced. Reduced ferredoxin acts as electron carrier and donate electrons to Fe ion protein to reduce it. The electrons released from ferredoxin are thus oxidized. And the reduced iron protein combines with ATP in the presence of magnesium and it donates electrons to nitrogen to ammonia. The enzyme set free after complete reduction of nitrogen to ammonia. So nitrogenase actually consists of two proteins that are work in tandem, the iron protein and molybdenum protein, as I have said you earlier. And during the catalytic reductions of dinitrogen, the electrons are transferred from Fe protein to molybdenum protein. In molybdenum iron protein, nitrogen is converted to ammonia, and in reductase Fe protein, ATP is reduced to ADP. So nitrogenase is extremely sensitive to oxygen, and root nodules of the nitrogen fixing plants contain the oxygen binding protein like hemoglobin, which protects nitrogenase by binding molecular oxygen. So here what is the role of like hemoglobin? It gives pink color to active nodules and regulates oxygen concentration to protect nitrogenase, which is oxygen sensitive. And what are the benefits of rhizobium symbiosis is, it increases soil nitrogen fertility, it reduces need for chemical fertilizers, enhances crop yield and soil health, promotes sustainable agriculture. And the summary will be the rhizobium legume symbiosis is a classic example of mutualism and it plays a vital role in nitrogen cycling and sustainable agriculture. Thank you.